Good day, this is Professor Will, CBMD PhD. Today is Wednesday, April 14th, 2021. It is 12:41 and 59 seconds a.m. Eastern Standard Time. Today's research, protein channel. So let's hop to it here. Inside the protein channel that keeps bacteria alive. Date April 13, 2021. Source, Rockefeller University. Summary. A novel method for studying how one crucial membrane protein functions may pave the way for a new kind of broad-spectrum antibiotic. Almost all bacteria rely on the same emergency valves, protein channels that pop open under pressure, releasing a deluge of cell contents. It is a last-ditch effort, a fail-safe that prevents bacteria from exploding and dying when stretched to the limit. If we understood how those protein channels work, antibiotic drugs could be designed to open them on demand, draining a bacterium of its nutrients by exploiting a floodgate common to many species. But these channels are tricky to operate in the lab, and how precisely they open and close, passing through a subconducting state and ending in a desensitized state under the influence of mechanical forces, remains poorly understood. Researchers introduced a novel method to activate and visualize these channels, making it possible to explain their function. The findings shed light on key membrane proteins in bacteria, and the same method can be used to improve our understanding of similar channels in humans. Researchers were actually able to see the entire cycle of the protein channel, passing through a series of functional stages. Research has long focused upon MSCs, a protein embedded in bacterial membranes that opens in response to mechanical force. MSCs, proteins exist in a closed state, while resting in a thick membrane. Scientists once suspected that when fluid buildup causes a cell to swell and puts tension on the membrane, it stretches, so thin that its proteins protrude. Thrust into an unfamiliar environment, the protein channels snap open, releasing the contents of the cell and relieving pressure until the membrane returns to its original thickness and its channels slam shut. Researchers tested this theory over 5 years ago, reconstituting MSCs proteins into small custom-designed membrane patches. They discovered that it was impossible to prise the channel open by thinning membranes within the natural range. They realized that membrane thinning is not how these channels open. These custom patches, called nanodiscs, allow researchers to study proteins in an essentially native membrane environment and to visualize them with cryo-electron microscopy. Researchers resolved to push the limits of nanodisc technology, removing membrane lipids with cyclodextrin, a chemical used to excise cholesterol from cell cultures. This induced tension in the membrane. The researchers could now observe with cryo-electron microscopy as the channel reacted accordingly, eventually snapping closed for good, a phenomenon known as desensitization. What they observed matched computer simulations, and a new model for the function of MSCs emerged. When fluids build up inside the cell, they found lipids are called in from all corners to help ease tensions throughout the membrane. If the situation becomes dire, even lipids associated with the MSCs channels flee. Without lipids keeping them closed, the channels have the legroom to pop open. They can see that when you expose the membrane to cyclodextrin, the channels open and then close again. Researchers' new method of manipulating nanodiscs with cyclodextrin will allow researchers studying dozens of similar mechanosensitive protein channels to, at long last, test our hypothesis in the lab. Many such proteins play key roles in humans, from hearing and sense of touch to the regulation of blood pressure. Of more immediate interest, however, is the prospect of exploiting protein channels that many different bacteria rely upon to survive. Novel drug targets are a particular necessity, given the rise of dangerous antibiotic-resistant bacteria such as MRSA. MSCs and the related bacterial protein channel MSCL are extremely interesting drug targets. Almost every bacterium has one of these proteins. Because these channels are so widely distributed, a drug that targets MSCs or MSCL could become a broad-spectrum antibiotic. Good day, this is Professor Will, CBMD PhD, on microbiology, bioinformatics, and pathophysiology. This video lecture was created for educational purposes only. Well, hope you enjoyed listening. Until next time, have an awesome day.